All right, let's go ahead and get started. So it's a real pleasure to welcome our first speaker today, Professor Kamil Godula. Kamil started his studies in the US, although he's from the Czech Republic, at Marquette University. And then he earned his PhD at Columbia University doing some CH activation chemistry. Camille entered the glyco world as a postdoc in Carolyn Bertozzi's group, where he has stayed, luckily, doing some fantastic science. He started as an assistant professor at UCSD and then won basically every assistant professor award you can win. So the Sloan, the Cottrell, the NIH DP2 New Investigator, and made tenure a couple of years ago. So go ahead, and we're looking forward to your talk, Camille.
So in the next 30 minutes, I would like to give you a little bit of little overview of what we do in our lab and zero in on one particular story that we just finished. And this will be the first time I'll be speaking about it. So I hope that that I make it through it without problems and that you will enjoy it. So my talk is focused on the design of glycomaterials that we use to try to engineer glycan interactions at cell surfaces to control their cellular activities, and particularly in differentiation. And so the area of the cell that we are the most interested in is the glycocalyx. So that's the ensemble of glycolipids and glycoproteins on the cell surface, as I show it here in my little cartoon, where all of these different glycoconjugates perform a variety of different functions involved in interacting with other cells, with proteins and the extracellular matrix, or in regulating cellular processes downstream of the membrane. And I'll focus on this particular area here, highlighted in red, which focuses on how certain glycoconjugates called proteoglycans may get involved in engineering of regulating sensing of biochemical cues, including growth factors, which then triggers processes downstream and cellular differentiation. And our approaches in our lab that we use to regulate these processes. Okay, so we take an approach that's based on materials. We sort of think of glycoproteins and proteoglycans in particular as a large biomaterial, and we are trying to mimic them in synthetic analogs, where we can control both the glycan composition and their interactions with proteins of interest. And we assemble these new materials, which we call neoglycoconjugates. They're really glycopolymers, and I'll show you the structures in just a second, on synthetic scaffolds, to which we then attach a variety of different sugar structures that we typically isolate from biological sources, but they can also be made synthetically. And because these materials are synthetic, we can also introduce additional functionality. So we can include anchoring elements. Those could be reactive groups that allow us to attach these materials to various different solid surfaces, where we can interrogate their interactions with proteins of interest. Or there can be lipids, and if there are lipids, we can then insert them directly into the cell membrane and thus introduce a new glycan interaction to the cell surface. And oftentimes we also introduce optical probes so that we can visualize and track these materials. So this is the sort of basic approach, or one of the basic approaches that we use in our lab to introduce and tune glycan interactions on cell surfaces. So in particular, we're interested in doing that in the context of signaling that regulates cell differentiation. So for instance, proteoglycans, as I already showed in the initial cartoon, are important for formation of complexes between growth factors and their receptors to trigger downstream signaling. So proteoglycans, which carry sulfated polysaccharides called heparin sulfates, glycosaminoglycans, or HS-GAGs, can modulate the association of growth factors such as fibroblast growth factor 2 with its receptor. And if this is in a mouse embryonic stem cell, that triggers a MAPK signaling pathway, which can be detected by phosphorylation of some of the kinases downstream, such as ERK1 or 2 kinases. And eventually this leads the cell to differentiate towards neural type lineages. If you delete this polysaccharide from the cell surface by, for instance, mutating out one of the key enzymes involved in assembly of these polysaccharides, all of a sudden the cells cannot engage the growth factors. The growth factors do not associate strongly enough with their receptors, and that silences this pathway. And if you can't activate the pathway, well, the cells will remain in an undifferentiated state, and they'll basically be stuck in that state. So our thinking was that perhaps we can borrow some of these motifs that you will find in these long polysaccharides and introduce them into synthetic materials, and those materials would then have growth factor binding activity. And if we can put these materials back onto the cell surface of these mouse embryonic stem cells that were mutated out not to produce their own polysaccharides, maybe we can reactivate these pathways. And if we can do that selectively, then maybe we can reactivate different signaling pathways that are associated with different types of differentiation. And maybe we can guide cells to differentiate in the direction that we want them to. So this project started, actually, this is one of the oldest projects in our lab, and it was started by a postdoc back then, Mia Huang, who is now a professor at Scripps, Florida, and she was helped by another postdoc, Alex Smith, and then first-year graduate student Greg Trieger, who has now also graduated and with his doctorate. And so, just to kind of show you how we did this, we first looked at the structure of the heparin sulfate polysaccharide. And I'm sorry, I'm moving things around because my Zoom control panel got in the way. Okay, so this is the polysaccharide, and it's attached typically to a protein, and that makes it a proteoglycan, this whole conjugate. And there's a lot of different enzymes that are involved in the biosynthesis of this polysaccharide. But in general, the polysaccharide is formed through polymerization, and then it is elaborated with a variety of different enzymes that introduce sulfate groups. Those could be enzymes such as NDST1 and NDST2, which will become important later on in my talk, which sort of initiate this process of sulfation by replacing acetamide groups on glucosamine with sulfates. And then that continues, and additional enzymes can introduce a variety of different sulfates on the 6th position of the sugars, or 2 position of the uronic acids, and so on and so on. And the long polysaccharides then will have dense regions that are highly sulfated, and those provide the binding motifs for the growth factors and also the receptors that are of interest to us. And here again is the XT1 gene that I introduced on the previous slide, which basically is one of the enzymes responsible for polymerization of the GAG. And if you knock out this enzyme, the GAG will not get polymerized, and you will not have activity at the cell surface. So here is a chemical structure that represents portions of these sulfated domains. And you can see that this is a very complex oligosaccharide, tetrasaccharide, and oftentimes these regions are much longer than that. And they're very difficult to generate synthetically, even though some of the more modern and recent methods in chemoenzymatic synthesis and also chemical synthesis have now enabled access to increasingly complex structures, such as this one. But when we started, synthesis of oligosaccharides of heparin sulfate was still very challenging, and we did not have expertise in that area in my lab. And so we decided that what we could do is we could use enzymes to break down these complex structures into the individual disaccharide building blocks. And what's nice about the enzymatic depolymerization is it doesn't really touch the sulfation patterns on these disaccharides. And though these disaccharides can then be separated out and isolated, and they're actually commercially available as standards for analysis of the composition of glycosaminoglycans. So we had ready access to these disaccharides. And those would be the building blocks for our materials. And we were thinking that if we could make materials with disaccharides representing these different types of sulfation patterns that you will find in the heparin sulfate, we can maybe get selective materials that can engage growth factors of interest. Okay, so in order to use these disaccharides, we needed a chemistry to attach them to the polymer scaffolds. And so we relied on a method that's oftentimes used to modify reducing sugars, and that is the Oxime Ligation. And you can take a nucleophile, such as this alpha heteroatom nucleophile, amino oxy group, and they have the ability to interact, to react with the reducing end, which is really a masked aldehyde, to form stable structures where the nitrogen of the amino oxy group is attached to the reducing of the sugar. To synthesize these polymers, a little bit of chemistry here, we used controlled radical polymerization techniques called the Raft method. And here we take monomers where we have our amino oxy group suitably protected to survive the polymerization process. And we polymerize them in the presence of a chain transfer agent and a radical initiator. And we do that, and adding this chain transfer agent allows us to control the polymerization in a way that then the polymer chains that we generate have the same or roughly the same length. And we can control that length. And within the population, the polymers look very much the same. And another advantage of using this polymerization is that the chain transfer agent ends up capping both ends of the polymer. And so this is a really great opportunity to introduce our anchors, which are shown here in the orange circle. And we can attach a variety of different groups. We can use biotin, which interacts very strongly with streptavidin. We can use a variety of different reactive handles, azides, propargyl groups, allyl groups for click chemistries. Or if we want to deliver these materials eventually to the cell surface, we just attach a lipid. Okay? Then in a couple easy steps or simple steps, we transform these precursor backbones into our reactive intermediate. So first, what we do is we remove the trithiocarbonate group using a base, nucleophilic base, and that releases a sulfhydryl, which we can then cap with maleimide fluorophores. And finally, we remove the Bach protecting groups to release our reactive amino oxy side chains. And we're ready to assemble our glycopolymers. And this is done actually under very simple conditions. We take our reducing disaccharide here, and we combine it with our backbone, typically under acidic conditions and acetate buffer, and we heat this reaction at 50 degrees for about 2 days or so. And at the end of the day, the second day, I must say, we have a glycopolymer, and we can purify it easily based on size by just spin filtering away the unreacted sugar. And thus we can generate an entire library of structures in parallel in a very rapid way. And each polymer in our library now carries a different type of disaccharide, or we also sometimes introduce monosaccharides as controls. And then we attach these polymers onto glass slides that were modified with cyclooctane. And so they will covalently attach, and you can visualize them by looking at the fluorophore label on the polymer. So the green dots represent the polymers, and each dot represents a different type of sugar attached to the polymer backbone. And now we can take this array and we can probe it with growth factors such as FGF2, which is of interest to us. And FGF2 is right here. And you can see that some of the polymers, but not all, will engage this growth factor. And when we take a look at the composition of the sugars, what we see is that we typically see binding of the growth factor to these polymers if we have a disulfate present in the molecule. So sulfate on this uronic acid and a sulfate on this amino sugar, glucosamine. There might be some modifications to the nitrogen, but in general, these two sulfates are required for binding of the growth factor. If you see here, there's a monosaccharide that's sulfated and then doesn't bind. And also disaccharides that don't have the sulfates do not bind the growth factor very well when they're arranged on the polymer. So there's some selectivity that's associated with the glycan structure, which is great for us. Now, we can take the winners and losers from our screen, excuse me, and instead of an azide, we introduce our lipid. And as you know, like dissolves like, and so the lipids drive the polymer from solution into the lipid membrane of the cells. So here we are looking at cells. Those are the mutant cells, EXT1 mutant cells of mouse embryonic stem cells that do not produce their own heparin sulfate, but they can be remodeled with our polymers. And the green here is coming from the fluorescent label. In this particular case, it's the LXL4 488. And I'm showing you examples of the two polymers. So this one would be the D2A6, that's the disaccharide shown here. That's the one that bound to FGF2 in the microarray. And this one is the monosaccharide and acetylglucosamine 6 sulfate, which did not engage the growth factor. So we can bring these polymers to the cell surface, and then we can test whether the cells will actually sense the growth factor of interest. So when we stimulate our cells with FGF2, normally because they don't have HS, we would not see activation of the MAPK pathway. And you can see that there's not much of an increase in phosphorylation of the ERK1 kinases after stimulation with FGF2. But here, this is our polymer that bound to the FGF2 in the microarray. We can see a robust reactivation of the signaling pathway. And here's our monosaccharide, and we also included the disaccharide polymer that didn't have sulfates, and it was also a negative control in our array. And typically what we do is we add heparin, which is a highly sulfated version of heparin sulfate, and you probably know it as an anticoagulant. And so when you add heparin into the media to these cells, it can substitute in some ways the cell surface HS polysaccharides and also activate the signaling pathway. So this was really our positive control. Okay, but in this case, the heparin is soluble; it's not attached to the cell surface. Okay, so that's great. We're able to reactivate the signaling pathway, and the question was, can we actually guide these cells now to differentiate? And in fact, if you take our remodeled cells and you subject them to neural differentiation, what you will see is after 6 days is an appearance of these new beautiful structures. These are called neural rosettes, and they result from exit of these embryonic stem cells towards differentiation. And under these particular conditions, you will get neural differentiations. And these are all of the stem cells, neural stem cells that will eventually go on to differentiate into different cell types of the nervous system. Here's a control colony. So these cells are the mutants that can signal through FGF2 because they do not have their native HS. And you see that they're still largely green, and the green represents a differentiation, represents an embryonic marker, OCT4. So you can see that they're not very good at exiting and differentiating. And the same is true for our negative controls here. So the polymers that did not bind to the FGF2 and did not activate FGF2 pathway, associated pathway, will not lead to differentiation. And by tuning in the amount of the polymer on the surface, we can tune in the number, or we can control the number of these rosette structures that we generate during differentiation. So not only we were able to turn on the signaling, but we were also able to tune it, the extent of it, by changing the density of the polymer on the cell surface. Okay, so this is to kind of introduce the method. This work has been published quite a bit ago, but what I would like to do is share with you some new data that we just from a study that we just completed, and so far it's unpublished, but we'll be depositing the work on bioarchives in the next few days. So you guys will hear about it before it first appears out there today. And it involves regulation of adipogenesis or adipogenic jet differentiation in the context of potentially a new way to address type 2 diabetes. So adipose tissues or fat tissues are really important in regulating glucose levels in the blood. The adipose tissue can expand if you are under high carbohydrate diet, and it can take up glucose from the bloodstream and store it as a fat. And that's a good process until you get to the stage when the fat tissue is too extensive and will give rise to inflammation. We can eventually lead to or contribute to loss of insulin sensitivity in the tissue or insulin resistance. So you can either get decrease in production of insulin, or you can lose receptors in the adipose tissues that can sense insulin. And in either case, you will affect the ability of the tissue to uptake glucose, since insulin is involved in regulation of glucose uptake. And when that happens, you're faced with type 2 diabetes, okay? So acquired diabetes. And today, the treatment for diabetes is really just by modulating glucose uptake. So you either need to decrease the level of carbohydrate that you take up in a diet, or undergo gastric bypass surgery, for instance, right, to limit the food intake. You can take appetite suppressants that still regulate the uptake, or you need to start taking insulin to replace the insulin that's not being produced in your pancreas. However, this increasing the amount of insulin requires that you actually have the receptors that sense the insulin. And so once you lose insulin sensitivity, that becomes a very big problem in the treatment of diabetes. So we started thinking that maybe another approach to help with treatment of diabetes would be to, in general, increase the amount of glucose clearance in an insulin-independent way by the adipose tissues. And the adipose tissues continuously expand, and new fat cells can be differentiating. And so potentially we could maybe change the way that they differentiate to produce adipocytes that are better at taking up glucose in the presence or absence of insulin activity. So this work started really by an important observation by my colleague Philip Gortz here at the Glycobiology Research and Training Center, when they looked at the formation of adipocytes from mouse embryonic fibroblasts. And Philip has a mouse model where you can conditionally knock out one of the genes involved in heparan sulfate sulfation, so NDST1. That's the enzyme that I sort of pointed out, which starts the process of sulfate, formation of sulfated regions in heparan sulfate. And so what he saw in his lab, that if you have an NDST1 deficient mouse, the adipocytes that are produced during differentiation from mouse embryonic fibroblasts actually lose ability to accumulate lipids. And so this is where my graduate student teamed up with Philip, and he was really interested in exploring this phenomenon. But we didn't really quite know where this effect was coming from. All we saw was that if you differentiate these adipocytes and then you introduce a lipophilic diol, oil red, you will see that if you don't have fully sulfated heparan sulfate, you store less lipid. Okay? And if you add heparin, the heparin can reduce lipid storage. If you add heparin during the differentiation, and the heparin can act as an inhibitor or antagonist of HS activity here, you also lose lipid storage. But if you add heparin to the NDST1 mouse embryonic fibroblast, it doesn't further decrease or improve lipid storage. Okay? So heparin here actually acts in a different way than it did in the embryonic stem cell case, where it could rescue the activity of heparan sulfate. But here, it acts as an antagonist of heparan sulfate or inhibitor. Okay, so that was a really interesting observation. So first, what we wanted to do is we wanted to figure out at which point do we lose the ability to store lipids, and whether it has something to do with maybe the ability of the adipocytes after they differentiate to take up fatty acids. And so we didn't really see a negative impact on fatty acid uptake, as you can see here, in the adipocytes that were either coming from the NDST1 mutants or that were treated with heparin. And we also didn't see much of a difference in the binding of very low density lipoproteins to these cells, if they liked HS or if they were antagonized with the soluble heparin. Okay, so it then didn't seem to have anything to do with the ability of the cells to take up lipids. But what we found out was that it was actually related to glucose uptake. So what we saw was that in the NDST1 negative cells, we reduced the ability to take up glucose. So this is a radioactive glucose uptake assay, and so the cells were taking up much less glucose. If we stimulated with insulin, we saw increase in glucose uptake, but but there was still much less glucose being taken up by these cells in the NDST1 negative cells. Okay, so something happened, and the cells do not take as much glucose, but they respond normally to insulin. Well, maybe it has something to do with the number of receptors that transport glucose from the outside to the inside of the cell, and we looked at expression of various different glucose receptors, and here's GLUT4, glucose receptor 4, and we don't really see much of a difference in the HS inhibited cells. So it doesn't have anything to do with lipid uptake, or it doesn't have anything to do with glucose receptors. Okay, and so then we looked at what happens to the glucose. So we observed that we actually get much less lactate in the media where we have HS inhibition. Okay, so the wild type cells produce quite a bit of lactate, the media turn yellow, but these HS inhibited cells, NDST1 or wild types that were treated with heparin, produce much less lactate. So that suggested that there's something going on with metabolism of glucose. And what we observed also was that the cells had overall higher levels of palmitate, which is a fatty acid, right, than the HS inhibited cells. So there's less lipid, and that makes sense because there's less lipid accumulation. And so that suggested that maybe if we inhibit HS during differentiation of these adipocytes, we are jump-starting the metabolism of these lipids, right? We are increasing the amount of lipids that are eventually being metabolized, and we're switching from glycolysis, which produces lactate, to fatty acid metabolism, which would then produce more citrate. And you can see that here. So if we trace the palmitate, we see that we actually get more citrate production compared to the wild type cells. So something happens during the differentiation. If the HS is not functional or it's inhibited, something happens that then changes the metabolic profile of these adipocytes after differentiation, and they take up more glucose, and they have a higher level of glycolysis compared to the inhibited cells. So now we need to figure out when this happens. When is the window in those 6 days of differentiation where HS makes difference in setting of the metabolic profile of the differentiated cells? So we did a couple different assays. We first introduced the heparin on day 0, and we treated the cells for different amount of time. So the differentiation always 6 days, but we withdraw the heparin at increasingly shorter periods. And so what we found out is that we needed the heparin to be present at least 3 days to see differences in glucose uptake compared to the untreated cells. Okay, now we needed at least 3-day treatment. Does it matter when the 3 days occur in the 6-day period? So we did the opposite of this assay, or we swapped the things around a little bit, and we started to introduce heparin at different time points. And what we found out is that we needed the heparin to be present on between day 1 and day 3. So 3 days, and it had to be the first 3 days of differentiation. And that's where the heparin sulfate on the cell surface is involved in regulating the metabolic activity of the adipocytes after they differentiate. Okay, so with narrowing down the window of opportunity here, we started to look at what sort of pathways might be involved in this process. And there's a lot of different growth factor signaling pathways and other signaling pathways that are involved, in which HS might be involved. And so we looked at mRNA, and what we did is we compared both the NDST1 cells and the wild type cells inhibited with heparin, and on an early day 3, and also later in the differentiation, to be able to tease out the changes that are really responsible for, or might be responsible for the change in metabolic profile in the adipocytes at the end of differentiation. And there's a alphabet soup here of various different genes that popped up. And so when we analyzed them, we realized that we are seeing what looks like changes in Wnt signaling activity. Okay, a lot of the genes that we saw being upregulated or downregulated were actually associated with Wnt signaling. And so to test whether Wnt activity was at play there, we use small molecules of Wnt signaling inhibitors. We use various different ones, but I'm showing you niclosamide here. And what you could see is that dosing in the Wnt inhibitor niclosamide was able to improve glucose uptake in the NDST1 negative cells. So, but we didn't see much of an effect in the wild type cells. So that suggests to us that the Wnt signaling activity is enhanced in NDST1 cells and also in the presence of heparin. And then we don't get much of a, and it's inhibited in the wild types. And adding the inhibitor doesn't really add much more to that. So this is interesting. So heparin sulfate is known to be involved in Wnt regulation. And it's been proposed to inhibit Wnt signaling by binding the Wnt proteins and sequestering them away from their receptor, frizzled. And so our idea was that maybe we can do that by using synthetic materials that have affinity for Wnt. And so we generated our mimetic polymers and introduced sugars that should bind to Wnt. So particularly this trisulfated disaccharide and this disulfated disaccharides. And we also used a non-sulfated disaccharide control. And the idea was that if we put this polymer on the cell surface, we could bind to Wnt proteins and pull them away from the receptors. This should inhibit Wnt activity and eventually lead to improved glucose uptake in these cells. And so first we double checked that our polymers could indeed bind to Wnt, and we tested at least two proteins, Wnt5A and Wnt10B, known to be involved in adipogenesis. And you can see the polymers bind to, here's showing the polymer with the sulfate groups, and it binds to both proteins. And then we performed a differentiation in cells that were remodeled with this polymer, actually with all three polymers. And again, I'm trying to move my video screen here, but I can't. Anyway, so what you could see here is that the polymers, particularly the sulfated ones, were able to improve glucose uptake in adipocytes, differentiate from these NDST1 cells. So they were actually able to reverse the activity of the deletion of the NDST1 enzyme. But even more importantly, which was really exciting to us, we saw that we also improved the glucose uptake ability of the wild type cells. And as you remember on the previous slide, I was showing you that Wnt inhibitors were not able to really improve glucose uptake. And that's because we were not able to dose them high enough without them having actual toxicity effects on these cells. So within the dosing range for niclosamide, we were not able to improve the natural ability of the cells to take up glucose. But our polymer strategy was actually able to increase glucose capacity in wild-type adipocytes by about 50% almost. So this is fantastic because this suggests that what we could potentially do by using these materials is to just, in general, influence differentiation of wild-type cells into new fat cells. And when they differentiate, they will be able to take up glucose. And if you remember, the glucose uptake capacity increased in both cases if you had insulin or did not. So it just basically raises the basal level of glucose uptake in an insulin-independent way, which may potentially provide a new approach for treatment of T2 diabetes or management by generally increasing the glucose clearance capacity of the fat tissue. Okay, so this is the newest story from the lab, and I'm so excited to share it with you today. And I think I'm out of time. And just to summarize, our recent findings show that cell surface heparin sulfate regulates Wnt signaling activity in an early stages of adipogenesis. And in fact, it actually downregulates Wnt by sequestering them away from their receptors. And that changes the metabolic activity of the adipocytes after differentiation. And the cells have a generally increased level of glucose uptake. And we're able to use our engineering approach, which is a simple addition of polymers with growth factor binding activity to the cell surface to alter the metabolic profile of cells, including wild type adipocytes. And again, this might be a new entry into the potential ways to treat type 2 diabetes by generally improving the ability of the tissue, the fat tissue, to take up glucose and process it. So with that, I would like to thank my group. I'm highlighting here the graduate student, Greg Trieger, who led the study. So he was a student in my lab, and also Mia and Alex, who originated the project, who were at the beginning of the project, focused on engineering stem cells with our glycomimetics. And so this is my lab at our annual San Diego Glycobiology Symposium, which is organized by the Glycobiology Research and Training Center. And these are my colleagues there, Philip Gortz and Jeff Esco, who are the HS gurus and glycosaminoglycan gurus at GRTC, and really wonderful friends and collaborators. And so once again, Philip is the collaborator with me on this project focused on adipogenesis that Greg worked with. So with that, I would like to thank you for your attention. I'll be happy to answer questions. I hope I'm not too much over time.